DNA 101.
Transcription happens in the nucleus when the DNA is unzipped by the helicase. It consists of G's, C's, A's, and T's and will eventually create mRNA.
Translation happens in the cytoplasm and nucleus and is the transition of mRNA to tRNA. It consists of G's, C's, A's, and U's, and the codons create tRNA.
DNA replication. The DNA starts as one strand, then the helicase breaks the hydrogen bonds apart. Then the primase synthesizes a short RNA segment, also known as the primer. The primer provides the starting point for DNA synthesis. Moving from the 5' to 3' direction, they build base pairs side to side. Polymerase builds the two main strands known as the leading and lagging strands. The leading strand moves from the 5' to 3' direction, and the lagging strand moves from 3' to 5' direction. The Okazaki fragments are used as a building block for synthesis of DNA in the lagging strand. At the end, the ligase seals it all together by gluing it.
Extraction of DNA from the strawberry lab. To begin our lab, our first step was adding 10 milliliters of dish soap and 1/4 teaspoon of salt to our beaker. This breaks open the lipids of the cell membrane.
After the solutions were measured, we completed the rest of the strawberry smashing procedure. We then drained out the liquid part of the strawberry solution mixture into another beaker. We were then given 5 milliliters of frozen ethanol to bring the DNA to the surface of the solution. From this, we extracted the DNA using a pipette.
Restriction enzymes, also known as endonuclease, cuts DNA in a specific sequence at specific areas, which are known as restriction sites.
PCR allows DNA to make extra copies of itself. It allows things like blood samples to be identified from each other when there is no obvious distinction. In our crime scene, Anna, the crime scene blood, and Dominique had the same blood characteristics, type AB blood. Person 1 had 40 BP, and person 2 had 10 BP. Since they all had the same type of blood, we used PCR to determine the difference between each blood sample.
DNA electrophoresis is used in testing DNA and blood from each other. In the Anna Garcia case, since we had two people match the type of blood at the crime scene, Anna and Dominique, we used DNA electrophoresis to determine whose blood it was at the crime scene.